Welcome, dear guests and viewers. I'm your host, Gabor Chodkowski-Dziuric, and I'd like to invite you to take part in the online COVID-19 International Conference of leading virology and immunology specialists. We are honored today to be hosts for Nobel Prize winners in the disciplines of medicine and chemistry, thought leaders within the scope of ongoing COVID-19 research, tracking and understanding its spread, or preventive treatment, as well as many other esteemed experts and specialists. While given the current circumstances, it's difficult to organize the kind of physical gathering we are all used to, we believe this live stream to be an important chance to hear the independent opinions of many leading experts on the most pressing questions concerning the COVID-19, one of the greatest challenges of the 21st century. Together, we might gain a better understanding of this very, very complex issue. For the past 9 months, the COVID-19 epidemic has been one of the most pressing problems plaguing the entire world. The virus has quickly spread on all continents, infecting millions. To this day, we don't fully know just how many people managed to recover from the disease and how many have succumbed to it. It truly might be the greatest catastrophe in modern history. As such, it is simply impossible to tackle a problem of such a scale alone. Thus, more than ever, we have to combine our knowledge, experience, and efforts to deal with this pandemic and its consequences. The virus knows no borders. And thus, we have gathered the foremost specialists and experts in the fields of virology and immunology across the world to join their forces together. The online conference has been made possible by the Foundation for the Support of International Projects and the IPF 2021 Global International Pedagogical Forum. Before we get started, I'd like to briefly remind you about the structure and schedule of the conference. Starting at 10 a.m. GMT+2 time, we will be joined by the first group of 15 participants. Each speaker will be provided 6 minutes to present their speech, followed by a half-hour Q&A session to discuss their findings with their colleagues. Afterwards, we will take a short break before gathering again with the next group of experts. And finally, it bears mentioning that based on the results of our shared work on this online international conference, we hope to establish a working group comprised of preeminent experts to tackle the issue of creating the necessary recommendations and instruments to resolve the global crisis created by COVID-19 pandemic, and perhaps together find the most effective treatment for this disease. So, without further ado, let's welcome our first group of experts. First up, we will meet with Dr. Sujatha Sunil, the principal investigator at the International Center for Genetic Engineering and Biotechnology in New Delhi, where she is leading a group on vector-borne diseases. Her research concerns primarily the vector-borne distribution, with a particular interest on vector-host pathogen interactions. Her work led to obtaining a wide knowledge on virus evolution, vector-host interactions, and host response, using high-performance omics platform and advanced molecular biology techniques. So, let's welcome Dr. Sunil. Dr. Sunil, do we have you online? Well, we seem to have a minor technical problem to begin with. Я уже вижу ее, но не слышу. И она нас тоже не. Уже вижу. Dr. Sunil will be talking about the epidemiology of SARS-CoV-2. We'll be talking about the epidemiology of SARS-CoV-2 and reinfection in India. And we have her online and just waiting for the final connection.
I can hear you. I can start if my friend.
Please then, the floor is yours, Professor Sunil.
Thank you. So I am Sujatha Sunil from the International Center for Genetic Engineering and Biotechnology. And I'm going to talk about the epidemiology of SARS-CoV-2 and the phenomenon of reinfection of this virus in India. As we talk, as of today, there are around, the active cases global, and the Indian cases clearly show that India is ranking second with the highest number of cases of COVID cases in the world. The infection fatality rate is about 1.89%, and the mortality rate says it's 1.8%, which could be an underestimate, but we need further in-depth studies and more surveillance programs to confirm these numbers. So if you're going to talk about the SARS-CoV-2 genome organization, it is a single-stranded positive RNA genome of about 30 kb long. It's amongst the largest in the category of the RNA viruses. And they are arranged in the order of the ORFs, which transcribe for the non-structural proteins, followed by the structural proteins, namely the spike, the envelope, the membrane, and the nucleocapsid proteins, and flanked by the 3' UTR and the 5' UTR on both sides. The mode of mechanism of infection of this virus is known. It's known to infect the type 2 pneumonocytes and ciliated bronchial epithelial cells, utilizing the AC as a receptor. So, owing to the number of cases that has happened since the first infection way back in January, there's been a humongous amount of data generated with respect to the kind of whole genomes that has been generated and the circulating viruses all across the world. And this shows that based on, it's a dynamic situation that is happening based on the mutations that are occurring in the various isolates across the world. And they have been clustered into 7 different clades across the world based on the mutation status. And amongst these clades, namely the G, GR, GH, which are derivatives of the G, apart from the S, L, V, and the others, GR clade seems to have the maximum global spread, followed by GH and G clades. So coming to the phenomena of reinfection in India, so we first sought out that this concept or this phenomena in healthcare workers in India, who are the frontline warriors for fighting against COVID. And what we did see is they were testing positive after 3 months of the first incident, after they tested negative for RT-PCR. And they also witnessed an absence of the neutralizing antibodies post the first episode. And whole genome sequencing of these viruses from both the episodes were taken and was done. And the sequencing information led us to confirm that it was indeed an reinfection and not an prolonged infection of the same episode. This was done as a case series in 4 healthcare workers. So these are the details of the samples where you've got 4 patients, namely the patient A, B, C, and D, and their follow-up samples, where the duration between the first and the second episode ranged from 19 days to 66 days. I do see that 19 days could be too less a time, but were to be considered as a reinfection. But what we do see is that the kind of mutations that was present in both the episodes led us to believe that they also could be a case of reinfection and not an prolonged shedding of the virus. This is the status of the phylogenetic analysis and the various mutation analysis that was done in the 8 samples that were taken, the 4 paired samples. And what we do see is that there were distinct mutations in the viruses from the 2 episodes, with new mutations observed in either of the episodes. And there were unique mutations present in each of the patients that were not present as part of the existing clade information that we already have that is circulating in India. Further, we also try to do a protein-based annotation for understanding the kind of mutation that were present amongst these paired samples for these 4 patients. And we also see that there is some heterogeneity of mutations within the paired samples of the patients, further validating that what the source of the virus for the 2 infections for the 2 times that the patients were infected were separate incidences or different sources rather than an continued infection program present in the virus because of the virus. So to conclude, what we do see is that the frontline healthcare workers are at a risk to constant exposure to SARS-CoV-2. Reinfections of SARS-CoV-2 is indeed a reality, though rare at present, based on the information that we have right now. And whole genome sequencing confirms these reinfections. Thank you for your time.
No, we thank you, Dr. Sunil, and here's hoping we'll have a chance to talk a little more about it during the Q&A section later on. Next up, we'll be talking to Dr. Giulio Tarro, the research associate at the National Research Institute and the National Cancer Institute in the US. Regrettably, we don't really have time to mention even half the awards Dr. Tarro was awarded, so we'll just mention the most famous one, the most famous achievement of his, discovering the so-called dark evil of Naples by isolating a respiratory syncytial virus in children with bronchiolitis. Can we welcome Dr. Tarro? Well, while we're waiting, I'll mention that Dr. Tarro will be talking about different background of the immunological response in accordance with the previous zoonotic experience with coronavirus.
Thank you, Mr. Chairman, ladies and gentlemen.
Welcome.
I'm going to speak. I was going to speak on different background on immunological response according to previous zoonotic experience with coronavirus. T cells were studied for their responses to structural, that is, nucleocapsid, and non-structural proteins, accessory like NSP7 and NSP13 and NORF1, of course, concerning the viral particles of SARS-CoV-2 in 24 subjects convalescent by COVID-19. CD4 and CD8 cells were recognized in all convalescents, which recognized multiple regions of the nucleocapsid protein. 23 patients recovered from the first SARS, it means that 2003, 17 years after the outbreak, still have a long-lasting memory of T cells with respect to the structural proteins of the nucleocapsid, which demonstrates a cross-reaction towards the nucleoprotein of the current SARS. The subjects without history of the primitive SARS in 50% of cases, 9 out of 18, have T cells that react with accessory proteins, that is, the structural NPS7 and others. Epitopic characterization of NSP7 specific T cells showed recognition of protein fragments with respect to animal beta coronaviruses, but very reduced for human common cold coronavirus. Therefore, infection with beta coronaviruses induces a strong and long-lasting immunity of T cells with respect to non-structural protein, and as recently published Nina Lambert and others from Singapore. The most comprehensive documentation on plasma therapy in COVID patients can be found in the article of K1 and others on the proceeding of the National Academy of Science. I want just to say a few words. The plasma transfusion has been successfully used in the other 2 coronavirus epidemics, SARS of 2002 and 3, and MERS of 2012 and 2015, entering the plasma at a precise stage of the disease. That is, when there is already poor oxygenation, and the patient is subjected to assisted ventilation with I would say, PAP helmet, but is not yet intubated. It is a therapy that, like many, presents a risk, but frankly, it is not clear exactly why the WHO made a delay in yielding their approval. Only in the case of serious disease for which there is no effective drug treatment. According to experience, especially from the University of Cambridge with Dr. Abraham Kappas. According to the experience of the first SARS and of the MERS, the children were not exposed to the civet cat and camels in a similar way. It was thought that the same fact could take place with the SARS from COVID-19. Indeed, children are infected with the virus without suffering a serious disease and represent an important source of infection. The virus is found in their rectal swabs. This was just published with my friends in Belo Horizonte in Journal of Pediatrics. Growing with age, many specific cells of the immune system are no longer active, and therefore the body loses its ability to respond effectively. In fact, it has been experimentally proven that young mice respond to lung tissue damage from viral infection through prostaglandins, while adult mice succumb. The juvenile immune system and its efficient T helper cells respond to SARS-CoV-2. The helper cells, especially CD4 lymphocytes, stimulate B cells to produce antibodies against the virus and control infection. In this case, Th2 lymphocytes are able to control the inflammatory response caused by the viral infection, preventing an exuberant and delayed reaction as occurs in adults. The different hormonal structure and the same prostaglandins favor the female subject against the coronavirus responsible for the current pandemic. I would like now finally to speak according a special puzzle that we can look at the lack of coronavirus outbreaks in Africa. The same crowded neighborhoods that would lead to the quick spread of other coronavirus may have protected the population from SARS-CoV-2. It is an hypothesis. Some level of pre-existing cross-protective immunity might explain why the epidemic didn't unfold the way it did in other parts of the world. Professor Shabir Madi said the protection might be much more intense in highly populated areas in African settings. It might explain why the majority on the continent have asymptomatic or mild infections. This was communicated shortly from the good friends of South Africa. I think with this good perspective, I would like to put my end. Thank you.
Thank you for your insight, Professor. And our next guest needs no introduction, but we'll try anyway. Professor Peter Doherty, the Nobel Prize winner for his research on specificity of the cell-mediated immune defense and currently patron of the eponymous Peter Doherty Institute for Infection and Immunity in Australia. Welcome, Professor Doherty. This segment will be slightly different than usual. Instead of coming with a pre-made presentation, we will take the opportunity to ask a series of questions to our preeminent expert. Professor Doherty, do we have you on the line? Well, while we are apparently waiting a few minutes, I'd like to remind everyone that while currently we'll have a series of ready questions to ask Professor Doherty, do remember that every participant of this group will not only be able to, but will be encouraged to take part in a discussion panel after we're done with all the presentations. So if you will have any questions to Professor Doherty or any other specialists we've invited, you are more than encouraged to ask them in about an hour. Well, it appears we might finally have connected. Professor Doherty, do we? No, sorry. It will take us a little while to connect with Professor Doherty. So in the meantime, we'll introduce our next guest, Professor Damian Purcell, also from Australia. Professor Purcell leads the molecular virology laboratory at the Institute of Infections of Immunity. A very established persona in the Australian scientific community, he's also the former president of the Australian Virological Society and an executive member of the Australian Centre for HIV and Hepatitis Virology. Welcome, Professor Purcell.
Thank you. So I'm at the Peter Doherty Institute for Infection and Immunity in Melbourne, and today I'm going to be talking to you about our research program into the SARS-CoV-2. Our institute is a new institute, 6 years old, and I want to show you how we connect research, education and public health, and particularly public health for genomics that informs back to our research. So in Australia, we've had 2 waves of coronavirus SARS-CoV-2 shown in this diagram here. And the first wave is mostly spread across all of the different states in the country. In blue is my state, which is Victoria, and the city of Melbourne has recently sustained the greatest level of new infections. So over the course of this infection, the outbreak, we've had 27,076 cases with 886 deaths. And the second wave, as I said, is entirely due to outbreak in Melbourne, which is a result of a breach in quarantine in a hotel, quarantine hotel, where returning travellers were housed for quarantine. Currently, we've kept this outbreak under control through strict public health measures. We're currently still in a strict lockdown, just coming out of curfew now that we have less than 20 cases per day. We have a very high rate of testing, 0.6% currently positive in Victoria. And it's this rapid early response and public health prioritisation that has made the difference. In Australia, where we sit in a pretty reasonable position in the neighbourhood of Korea, South Korea, Singapore, in terms of our total number of cases. So as you can see, the second wave has made some dent on that profile as we went into winter and now coming out of winter into summer. So our testing rate is high, as I said, 0.6% positive, and we've tested very, very greatly in the community. But the important thing is these tests, the diagnostic samples, have been linked to genomics, and many of these positive testing samples have been screened for full genome sequences, which gives us an opportunity to understand the mutations that the virus is undergoing. So many of you might be familiar with the D614G mutation in the spike. And we're particularly interested in mutations surrounding the spike, particularly in the receptor binding domain, that might actually increase transmission and cause potential problems for vaccines and antibodies targeting the spike protein. I think we all know now that the D614G increases transmissibility by increasing the exposure of the receptor binding domain. So with the deep level of sequencing that we've done, we've actually had witnessed a strong penetration of the D614G. So almost all of the new cases now have this, but I want to draw your attention to three particular receptor binding domain mutations, particularly occurring within the receptor binding motif. So even though we've measured many, many mutations, 115 different single amino acid or double amino acid occurrences across the whole spike, these ones are picked out because they had expansions, little clusters, N501Y, 34 cases, G485R, 40 cases. And then this one here, which was the hotel quarantine breaching virus that now is up to about 4,000 instances. And all of these occur within the receptor binding motif. If I move to the next slide, it takes a little bit of time to come up because it's got a lot of material on it. Oop, it's jumped over. Here it is, okay. So you can see here some of the story in the genomics of our outbreaks here in Melbourne. So in orange are the international travellers that were easily distinguished by unique motifs, so barcodes essentially within their sequence. And as these mutations emerged, you can see three different individual transmission clusters. The first one at the bottom here is the G485R. Next in green is the 477N with many individual connected clusters. And I lastly want to finish up telling you in a little bit more detail about this N501Y mutation, which occurs again within the receptor binding motif and 34 instances in Melbourne. Now this mutation was interesting because we noticed that a similar mutation had arisen in mice, where SARS-CoV-2 was adapted for replication in mice in China. Same mutation, and when we look across the database, this position has changed also in the Netherlands, N501T, and this was the outbreak in minks that was controlled by culling mink. One other instance in the United States, but otherwise a run of these mutations arising in Victoria, in Melbourne, with most of them essentially being identical. Importantly, they were different in terms of other locations of mutations throughout the genome compared to the mouse-adapted virus that arose in China. So we wondered whether this virus could in fact infect mice, so we made soluble and human and mouse ACE2, and we found that the index strain could be outcompeted for binding in the Vero cell infection assay by human, but not mouse ACE2. Whereas the new isolate, the N501Y, which occurs in conjunction with the 614G, could be competed for binding by the mouse ACE protein. And sure enough, this clinical isolate with no adaptation was able to replicate in Bell C and C57 Black 6 mice. The impact of this in terms of immunity is something that we're actually quite concerned about, because a couple of convalescent plasma from arisen in patients infected with the index strain, when tested with the isolate, we've made the isolate, we find that this virus is more difficult to neutralize with those sera. Another very dominant mutation, the 477N, which is now essentially ubiquitous in Australia at the moment, is perhaps more readily neutralized, but still has some degree of resistance compared to the wild type index Wuhan strain. So investments in genomics particularly have prepared us well for our current pandemic, and the diagnostic strategies that include full genome sequencing have led to a very useful tool, a mouse-adapted clinical isolate, or mouse infectious clinical isolate, I should say. And these mutations in spike are emerging. Some of them could have important implications for vaccines, therapeutic, neutralizing antibodies, and potentially also antivirals. But importantly, strict public health measures, as I showed you in the beginning, can control and extinguish these arising mutant strains. And when we turn to analysing vaccines as we are now, neutralising antibody breadth has to be considered in the context of these emerging mutations. I just thank my collaborators acknowledged on this slide here. Thank you.
Thank you, Professor. And now I believe we might have chance to finally connect with Professor Doherty in a minute.
I could have talked for longer.
Yeah, now we'll feel silly for rushing you. But I suppose it's not that easy to connect with literally the other hemisphere right in the morning. Oh, I do believe we are establishing a connection right as we speak. So please don't go away. Even though I'm afraid there might be some little hiccup in getting the professor on the line, as these things regrettably tend to happen as everyone forced to work through all sorts of Zoom meetings and other communicators, be it for the meetings, conferences, or everyday work, has grown to know. Yes, I'm afraid we are still waiting for confirmation whether we'll manage to connect with Professor Doherty right now, or we'll connect with one of the following guests and leave Professor for later. Hopefully, in a minute or two, the entire matter will be resolved. So even if we might be having some technical problems in that regard, please do not worry. Other than that, the schedule will be maintained, and one of the reasons we chose to implement breaks between each of the groups is that in the case of such a delay, we are able to extend the session a little longer. Although I do believe instead we are connected with Professor Anupam Varma. President of the World Society for Virology and former President of the Indian Virological Society, as well as an elected member of the Indian Academy of Sciences. Welcome, Professor Varma. Can you hear us, Professor? Because we can definitely see you, but not hear just yet. Professor Varma will be talking about the new standards and experiences to guide future pandemic management learned over the course of dealing with the COVID-19 pandemic. Such as strengthening the public health system, building rapid response capacity, and so on. Yes, while we have Professor Varma online, it appears there are some sound issues on his side of the connection, so let's hope we'll be able to resolve them immediately. Can you hear us, Professor Varma?
Hear me?
Oh yes, now we are hearing you, Professor. So please go ahead.
Oh, okay. Okay. So you want me to start talking to non-audience, but anyway, don't worry.
Oh,
Thank you very much.
I believe the audience is already hearing you. Basically, if I can hear you, the audience also can hear you.
Okay, no problem, no problem. Okay, thank you very much. Well, first of all, I would like to thank the organizers for giving me this opportunity to be a part of this important program. I'm going to briefly touch about the new standards and experience to guide in the management of future pandemics. All of us know that COVID-19 is the most devastating pandemic in living memory, causing over 1 million deaths, of which nearly 0.1 million in India alone. We can hardly afford such big losses. There are over 33 million people affected in the world with this pandemic. It has shattered the world economy. I must compliment all the countries for their basic response, including the basic preventive measures to stop the virus from spreading. In most of the country, it was done fairly quickly, but even that quick was not quick enough. I'll come back to that point later. The nations that followed the basic preventive measures have successfully contained the virus. And I would like to give here credit to New Zealand and Senegal for their management in two contrasting situations of resource availability and infrastructure. India, considering the size, population, and demography, has done very well. On March 9, India had only 44 cases. Of those 44 cases, 35 had come from Italy. The people going to Italy and coming back. The remaining were for 4 other countries. That was the time to stop it. It was tried. But those measures which I just said, which were put in place, they have to be implemented and followed by people for the success of any program on this kind. Our only hope at the moment is to get a vaccine. There are more than 300 vaccine candidates which are in various stages of development. We are very hopeful that by early 2021, we will have effective vaccines, a set of vaccines. And I tell you, this will in itself a remarkable feat of development, of cutting down time. It has never happened that before. Indian pharmaceutical companies are at forefront and developing effective products, but the task is enormous. The main task is of getting vaccines, especially to the countries with fewer resources, and also to arrange their distribution. India is preparing itself very well to do that, as you can feel that with 1.35 billion population, it's going to be a very difficult task, but it will be achieved. What we need to do is to build rapid response capacity, medical infrastructure. It was built, but it's not sufficient to control. Development of reliable and quick diagnostic system. Currently, we were not prepared. It took time in developing it, but now we have to be prepared for such emergencies. Efficient monitoring system, so that whatever the policies are in place, they are implemented. Traditional medicines have shown promise. People are, very happy with this in countries like India. But there's a need to develop scientific data, robust scientific data on the use of these medicines, especially for developing immunity in people or helping in building up immunity. What the world needs to prepare for is to have the capacity for monoclonal antibody production. Monoclonals are the ones which can provide the quick response in any eventuality of this kind. It's expensive, but I'm sure we can cut down on the cost by making it more efficient, productive system. Convalescent plasma therapy has been shown to show promise, but it has not been used that much as one would expect. I think whatever the conditions are, we have to overcome those difficulties and improve it. The challenges continue, which especially for opening of schools and colleges. Revival of research and education in all areas. Research on COVID has increased tremendously all over the world, but research in other areas has suffered. We cannot ignore that for long. Revival of economy. Cutting down on the time of production of vaccines. More importantly, as we see now these days, countering misinformation about COVID. Understanding the virus' mysterious consequences and the variability which is there in the viruses. Understanding the virus reservoirs in small animals, particularly bats and rodents, which play an important role, and in the future, they could be the source of such pandemics. We must accept that the new normal for us is physical distancing, mask wearing, hand washing, and etc., etc. I don't want to list that. Time is very short. To conclude, COVID-19 is here to stay. We have to live with it. The future depends on as to how quickly technologies are developed to contain the pandemic, because still it's increasing. We have not peaked in many countries. sustainable building of capacity to protect human, animal, and plant health from pests and pathogens that might emerge in the future. This is the message I would like to give. And with this, I thank you again and thank the organizers for arranging this.
Thank you, Professor Varma. And we're very glad to have this kind of perspective also on our conference, because while oftentimes it's easy to get lost in the search for the technological marvel of the cure to end the whole disease, in practice it is no less important to deal with the oftentimes boring or unglamorous logistics of healthcare to deal with a pandemic such as this.
Thank you very much. It has been a pleasure. This is just a highlight of the points. Thank you.
Thank you.
I hope I have not taken too much time.
Oh, don't worry about that.
Okay, thank you.
Yeah.
Thank you.
Now we'll be going.
Your voice is not coming yet, ma'am.
And now we'll be going to our next guest.
Okay, then.
Our next guest, whom I believe will be the Professor Alemka Markotic, Director of the Dr. Fran Mihaljevic Infectious Disease Clinic in Zagreb, Professor of Medicine at the University of Rijeka, and also an Associate Member of the Croatian Academy of Sciences and Arts. Welcome, Professor Markotic. Markotic.
Thank you very much.
I'm probably butchered your name. I'm very sorry for that.
This is okay. Well, can I start?
Of course, go ahead.
Yeah. Well, I would like to thank ladies and gentlemen, to organizer for the kind invitation, and I would try to give you a few hints about the management of COVID-19 in Croatia, based on traditional and modern approaches to one emerging infection like COVID-19 is. So the excellent example is if we take like personal protection equipment. So this is a medical doctor in 17th century, and they didn't know at the time even that the microorganisms exist. But obviously they realized if they completely covered and they have this stick for the social distance, they could handle at least partially with the patients who suffered at that time from the plague, from the smallpox or cholera. Now here we have completely protected modern medical doctors. So I ask you, who did better? I'm not sure. But looking in Croatia, we can probably say that we contributed a lot to the fight of infectious diseases during the history. So Croatia invented and established in Dubrovnik in 14th century the first quarantine on 27 July 1377. We also, and this is romantic view nowadays, we also have a long-term tradition in public health, and the School of Public Health was established in Zagreb in 1927 by Andrija Štampar, and Professor Andrija Štampar was actually the father of WHO and founder, and he was elected as the first president of the assembly anonymously, and he didn't even establish a strong support to the different public health measures 100 years ago almost in Croatia, but all over the world. But let's go back to COVID-19 and Croatia, and we can say that in the first wave and in the first phase at the very beginning of the outbreak, we had really good situation. We succeed to keep numbers low, number of infected people low, and even number of dead people. And in almost 2 months, we actually dropped down the numbers on very few or on zero. Well, on the other phase, at the middle of June, our tourist season started. So during the whole tourist season, we had about 10 million of tourists. And to remember you, Croatia has only 4 million residents. So for 14 million people hanging around in Croatia at 2 months, we actually still have good numbers, and we controlled the outbreak and have a significant drop now again in the number of cases. And looking in comparison to Europe and the world, we can say that except of the small period of time at the end of August and part of the September, when we were higher with the number of cumulative number of infected people above the European numbers, we usually have it in a proper way, lower than Europe or the world, and especially with the mortality rate, which is strongly below the EU or world numbers. So what was the Croatian way? It was early shutdown and initial activities already in January 2020. And we were among the first in Europe who established RT-PCR detection. Our hospital established that at the end, before the end of January, and helped actually to other institutions to establish that. So far we have more than 20 diagnostic laboratories all over the Croatia, and the first case we had on February 25. We have also a tradition of infectious diseases, hospitals, clinics, and departments. So the advance was that we actually hospitalized all our patients in our infectious diseases clinics and departments, and we didn't mix them with other like internal medicine departments or surgery. So we successfully keep them isolated and mostly didn't spread infection all over the different hospitals. We had all the time and still have strong and prompt collaboration between infectologists and epidemiologists. And although we had shut down, we put it appropriately and gradually introduce different epidemiological measures and try to communicate them each time very strongly with the population. We even faced the earthquake in the Zagreb, and you will see that we successfully managed that situation as well. So we would like also to underline that at the very beginning, good communication between experts, Croatian population, and media with strong political logistic support really helped us to manage COVID-19 in Croatia. Of course, we continue to do our research, and I will show here some of our findings on molecular diagnostics or molecular analysis of our samples, which we did recently together with Luka Cicin-Schein, which is Croatian scientist in Germany, and our hospital and their team found out different quasi-species in SARS-CoV-2 samples. So overall, our research has far-reaching implication for development of antiviral strategies, suggesting that viral quasi-species which we detected may facilitate rapid emergence of escape mutants under selection pressure, such as the treatment with antivirals against the new coronavirus. So we realized that in the future, we will probably need few different antivirals to fight at the same time with the new coronavirus. We also looked for the situation about the wider population, like industry population and serological response. And in the first phase in April, we had still pretty low percentages between like 1.20% up to 7% among different industry population. Looking for the success of serology and for the possibility to use it for detection of acute cases, we could see that, and we could conclude that we need to combine several different tests, like ELISA tests, like point of care tests, but still this is not the first choice method, as you all know, for the detection of acute cases. But if we combine different serological tests, we have in a part detect acute cases about day 5 to 7. As I mentioned at the beginning, we had earthquake in Zagreb at the middle of March, and we expected that we will maybe have more cases after that situation. This was situation in our hospital on a day of the earthquake, but we actually looked. This was day of the earthquake, and we followed the number of cases after that in Zagreb town, and we even found a drop. We didn't find an increase among the cases. So it was actually two very scary and very difficult situations, and we succeed to actually fight them with the old measures, social distance, hygiene, testings, and similar. So at the end, I would like to thank you for your attention, and this is our diagnostic team in our Hospital for Infectious Diseases. Thank you very much.
Thank you, Professor. And now it's time for our following guest, whom I believe should be Professor Damian Jean. Though, let's see. Yes.
So thank you for giving me this opportunity to present our work on combinatorial steroid and antivirus therapy for COVID-19 in the hamster model. So this is the life cycle of SARS-CoV-2 and the existing drugs that can be used. For example, ribavirin inhibit mRNA capping, Taiwan interferons that have antiviral and immunomodulatory activities, and protease inhibitors, and remdesivir that target the RNA polymerase and also the proofreading exoribonuclease. So if we compare these to HIV, there are many drugs that we can develop that target all the different steps in viral replication. For example, we might develop drugs that block entry, the protease inhibitor, replicase, and other enzymes such as helicase, and also cellular targets and inflammasome inhibitors. For example, we actually just published these So as we shown here, that these ranitidine can actually inhibit viral replication in CACO2 cells. And in the infection model of hamsters, we can also show the suppression of viral replication, both in nasal cavity and also in the lung. And then if we look at the pathology, I don't have the chance to show you the histology, but just the histology scores. Remdesivir and also this ranitidine have the same activity to relieve the inflammation in the lung and in the nasal tissue. And as we show here, that actually these ranitidine, an approved drug for bacteria, can actually inhibit the SARS-CoV-2 helicase. We show here that it actually inhibits both the ATPase activity and also the DNA unwinding activity with very tight kinetics. So for the treatment of COVID-19, we can use repurposing drugs, for example, Taiwan interferon, ribavirin protease inhibitor, as I said, and remdesivir steroid. But however, there is also an important question, which is, are the steroids doing more good than harm? Because there are actually a balance between antiviral and anti-inflammatory. The steroids can actually, at the same time, inhibit inflammation but promote viral replication. And there are lessons from SARS and MERS that the steroids are actually causing some harm. So for the anti-inflammatory drugs of COVID-19, interferons might be one candidate, and also these IL-6 inhibitors, they actually have anti-inflammatory activity. And for the remdesivir, it actually targets both the viral polymerase and also this proofing reading enzyme. However, in the literature, we know that the corticosteroids, either the dexamethasone and also the methylprednisolone, in this very old paper published in 1964, it was found that the corticosteroids can actually sensitize the mice to infection with mouse hepatitis virus, which is a typical coronavirus. And the effect could be from 50-fold to 1,000-fold. So we face this dilemma, whether we put antiviral or anti-inflammatory as the priority. Otherwise, we might be adding fuel to the fire or to pulling the plug so that the inflammation could stop. So we tried methylprednisolone with these human macrophages from the peripheral blood. And as you can see, we tried 3 different treatment schedule, either P treatment or treatment at 1 hour after infection or later. And we tried 3 different donors, but in these donors, all 3 treatments can actually sensitize the macrophage to infection. And for these donors, only a P treatment can do so. And for this one, the effect is less dramatic. However, we still believe strongly that actually these steroids can sensitize the human macrophages to infection with SARS-CoV-2. And if we use these directly on the hamsters, we have actually developed a hamster model for COVID-19 that can replicate that can capitulate the mild and moderate form of COVID-19. So if we give a steroid on day 2 or day 5 and then do the sampling, we can see that the body weight loss has been reversed. So if we use steroid, either an early treatment or a delayed treatment, both can have an effect on the reversal of the body weight. And then if we look at the viral load, if we look at day 14 days after infection, we can see that the viral load is still high, remain high. So that's the concern. But if we check for the immune response against the N protein, it's actually okay. There is no strong effect on the adaptive immunity, the antibody response. If we look at the histology, I can tell you that actually the inflammation has been relieved both at day 7 and day 14. If we use the steroid, methylprednisolone, if we do this as a monotherapy. But here I will show the benefit of a combination between the methylprednisolone and the remdesivir. So if we at the same time give remdesivir, we can see the reversal of the body weight change. The body weight loss is actually the same. However, if we look at the viral load in both the nostril and also the lung, we can see that actually with the addition of remdesivir, the viral load is actually being inhibited. And the antibody response, there is no significant change, and the inflammation is relieved. So as you can see here, that if we treat with steroid alone or remdesivir alone, or together with remdesivir together with steroid, then we can see the viral protein expression. So the viral protein expression is not reduced much if we treat with steroid alone. But if we treat the cells together with remdesivir and steroid, then in the lung tissue, we can see that we can get a much better suppressive effect on viral protein expression in the lungs of the hamsters. So to summarize these, methylprednisolone sensitized human macrophages to infection with SARS-CoV-2, and methylprednisolone reduced tissue inflammation and reverse body weight loss, but prolonged viral clearance. So if we use methylprednisolone together with remdesivir, we have similar suppressive effect on tissue inflammation and body weight loss, but at the same time, we can actually reduce viral loads. So combinational steroid plus antiviral should be tested in randomized clinical trials in humans. That's our conclusion. So thank you very much. These are our collaborators who actually are the first to isolate the virus and also to show human-human transmission in China. That's my colleagues and my collaborators. And these are the people in the lab, and the work is done by Dr. Ye here. Thank you very much.
Thank you, Professor. That was very interesting. And next up, we'll hear about research and development of COVID-19 medicine from Professor Anthony Kelleher, Director of the Kirby Institute at the University of South Wales in Sydney. Professor Kelleher is perhaps best known for making the first observations describing the recovery of antigen-specific CD4 plus T-cell responses in patients receiving antiretroviral therapy, more specifically the ritonavir. Welcome, Professor Kelleher. Oh no, sorry, there's been a change of plans, and now we have connected to Professor Jean-Claude Manuguerra, head of the Department of Emergency Biological Care at the Pasteur Institute in Paris, who will tell us about the resistance of viruses outside their hosts. Professor Manuguerra is best known for his work on flu and Ebola, work which has led him to the position of the head of the Scientific Council of the World Health Organization's Global Influenza Program. Welcome, Professor. Can you hear us, Professor Manuguerra?
Yes, very well, thank you.
Please take the floor then.
Okay, thank you very much. So first I want to thank the organizers for this online event, and I'm going to talk in my allowed 6 minutes about the resistance of viruses outside the host. So between an infected person and an uninfected person, the virus has to go through the environment where it needs to survive. The transmission of respiratory viruses, take for example influenza viruses, can occur through direct contact with an infected person or with contaminated surfaces, by inhaling droplets or bioaerosols, or by contact with contaminated water. But there's a continuum between water, droplets and aerosols and the air. Contaminated water can produce droplets and be aerosolized. Droplets can fall and contaminate surfaces, and water washing contaminated surfaces can end up contaminated. Several factors can then determine the persistence of this virus in the environment before complete loss of infectivity. Among them, the temperatures plays a central role in driving environmental persistence of the virus. In the air, the temperature is in the center of the circle, is probably the most important factor, and it also affects another factor which is important, the relative humidity. And then this can modulate the evaporation and thus the size of the aerosols. When human respiratory aerosols evaporate, then the environment of the virus changes as the salinity and the pH can vary. We can find virus in water, for example, where the sludge has a protective effect, and it has been shown that it can absorb the poliovirus and protect it against UV. So all this, the study of these different factors affecting virus persistence contributed greatly to the development of virus control methods and public health in the population, as it's summarized here in the circle. For example, we noticed that in some countries, especially temperate countries, the virus are very seasonal. For example, in France, in 2 seasons, influenza, between 2016 and 2018, the highest average weekly incidence of influenza per 100,000 inhabitants in the dark blue circle here occurred when the temperature was between -2 and +7 degrees Celsius, and when the absolute humidity was between 3 and 7%, and the relative humidity between 60 and 100%. So all these factors are important, apparently, for the circulation of the virus. The early studies on the viral persistence in the air date from the 1930s, long time ago. In 1934, Wells published this result of an experiment assessing the time required for liquid droplets varying in size to fall on the ground or to evaporate after being released from 2 meters above the ground. According to his results, which were obtained in the saturated air, liquid droplets having a diameter or little smaller than 0.5 millimeter will evaporate before reaching the ground. He also deduced from this data that in the same atmosphere, droplets with a diameter from 1 to 10 micrometer could stay in the air between 10 and 16 hours. And these results are still quoted today, like in the article of 2006. 8 years later, the same scientists published results of an epidemiological survey in schools where he tested the effects of the presence of ultraviolet radiation on outbreaks of varicella, measles, and mumps. The early study on the trajectory of the droplets on an aerosol can vary whether you breathe, normally cough or sneeze. For example, trajectories of droplets and aerosol from a patient who sneezes droplets will travel for 6 meters at a speed of 50 meters per second. When the person is coughing, then the droplet will travel much less at 2 meters and the speed of 10 meters per second. And when the person breathes normally, then the droplet will travel only to 1 meter distance with a low speed of 1 meter per second. This study shows how long SARS-CoV-2, now we come to this virus nowadays, can stay and survive in different environments. As you can see on the first column, in aerosol, the virus can be detected up to 3 hours. On non-porous surface like copper, it can be detected live up to 4 hours. On cardboard, up to 1 day. And then on certain surfaces like steel, it can be detected up to 3 days, and on plastic, up to 4 days, which is remarkable. And you can see here the comparison between SARS-CoV-2 and SARS-CoV-1, and the behavior is quite close from each other. Now, in our lab, we looked at virus inactivation in items manipulated in biomedical laboratories at 3 temperatures commonly used in these labs, such as 50 degrees Celsius for blood decomplementation. The virus is then inactivated in matters of minutes, up to 15 minutes in cell culture supernatant and sera at 56 degrees, and less than 10 minutes in nasopharyngeal samples, for example. But we can look for the virus also in the environment at large. Here is the detection of SARS-CoV virus RNA in wastewaters of south of France post-lockdown. What is interesting here is that here is the curve of the detection of the virus in wastewater cleaning station, and you can see that it rose very quickly in June here, whereas the number of new cases in human clustered and started to increase much later. And the idea, and we worked on it with the detection kit in the environment, it helps rescue services to detect a cluster before the first human cases appear. And this is a tool that probably will be very useful. So now to conclude, because I already reached 7 minutes, most studies dealing with SARS-CoV virus environment detect only virus genome, not its variability. And I think we should emphasize that most studies should focus on virus ability to grow, because it will show whether it's contagious or not. This is also a question not resolved for influenza, but the relative role of fomites, droplet, and aerosols in COVID-19 transmission is to be addressed, probably taking into account specific settings and epidemic phases. And then, of course, the big question of the efficacy of various masks and social distancing must be more extensively evaluated. Thank you very much.
Thank you, Professor. And now we're about to go to our following guest. So this time I won't be taking a guess who manages to connect to us. Though, oh, I believe we're getting a connection. Yes, let's welcome Professor K.M. Yacob, the Chief Medical Officer at the Varma Medical Center in India. Professor Yacob specializes in studying symptoms of various diseases, in particular the body temperature, which is exactly what he will be talking about right now. Welcome, Professor Yacob. Well, we have Professor Yacob on the line. We have his presentation ready, though I believe the audio-video connection is still getting configured. Professor Yacob, can you hear us? Regrettably, myself, I've grew to expect that this sort of issues with video conferencing will be as much a part of our everyday life as washing our hands and wearing masks for the foreseeable future. Oh, we might be getting a connection. Professor Yacob, are you with us? Well, thankfully we still have a few minutes to try and resolve this issue before having to go further with our conference. Oh, I do believe we are about to resolve this issue in just a moment. Well, should Professor Yacob prove unavailable due to technical reasons, I believe the following guest would be Professor Irja Lutsar from Estonia, the Professor of Medical Microbiology and Virology at the University of Tartu. At the moment, Professor Lutsar is actively engaged in the study of coronavirus, which is exactly what she will share today with us. Her speech about the scientific approach to combating COVID-19 that happens right now in Estonia. Oh, I'm afraid we might have stumbled on an unfortunate series of several participants suffering from technical issues. So right in a minute, we'll see who will be the first person who will manage to get online. As there still are many interesting speeches for us to hear today. And soon after connecting with a handful of participants left, we'll go to the Q&A session until 12 o'clock GMT plus 2 time. Yes, I believe I can already see our moderators establishing a connection right as we speak. Well, while we're still resolving the technical issues, I would like to take this opportunity to seed the question I would personally like to ask of you on the upcoming discussion panel that will happen in a few minutes after, I believe, about 2 other speeches, namely the question of lockdown measures to be implemented in the upcoming autumn flu season, as in the first outbreak of COVID-19 in spring, the quarantine and lockdown measures, introduction of strict social distancing proved rather successful in most countries, but nowadays, in late September, populations of many countries across the world are simply put tired of said measures. And in practice, it might be difficult to enforce a quarantine such strict as we've seen in May or April of this year. On the other hand, we've successfully managed to introduce a common mask-wearing regime in the Western countries. So the question I'd like to ask of you, and wait for the chance to hear back in a few minutes, would be, would such a mask regime prove enough should there be difficulties in reinforcing strict lockdown? And having asked that, we finally managed to connect with Professor Yacob to talk about the importance and role of body temperature measurements in COVID-19. Well, we had Dr. Yacob just a second ago, so let's hope I didn't jinx it. Dr. Yacob, can you hear us?
Yes.
Oh, welcome, Dr. Yacob. We have you on our stream. We'd like to thank you for taking your time to participate in our conference, and please take the floor.
Yes.
Yes, we can hear you, Professor.
Can you hear me?
Yep. We can hear you,
Yes.
and we can
Okay.
see your presentation. Please feel free to begin your speech.
Sorry. Okay.
Professor, are you still with us? Well, it's not always easy to connect despite the supposed quality of our technology nowadays, but I believe this particular speech will well be worth waiting for. I believe the exact problem at hand concerns the exact devices through which we are connected, and that professor is simply trying to switch to another one that would better integrate with the software. Professor Yacob, can you hear us
Yes.
again? Yes, please,
Yes.
please begin your presentation.
Okay. The importance of role of both. I am Yacob Matai. Sorry. I am Yacob Matai from India. Yes. I am Yacob Matai from India. Today I want to discuss with you the subject is the importance of the role of body temperature in COVID-19. In COVID-19, up to 80% of patients have no fever. Why up to 80% of patients with the COVID-19 virus have no fever? Is it because there is no need for the care of fever, or is it because the disease is free of signals or symptoms? Let us examine the present state of fever. One medical book says our understanding of the moral basis of thermoregulation and fever is still rudimentary. Different type of fever definitions are seen in various medical books. Okay. The following 7 questions need to be answered to know about the fever. What are the ingredients necessary to develop a fever? What are the ingredients necessary to curing a fever? What is the purpose of temperature of fever? The heat energy used for which activities in fever? What are the problems not knowing the purpose of fever? What are the difference between actual fever and temperature of fever? During fever, why our body acts against the effects of disease? No medical books give a clear answer to about 7 questions. Necessary ingredients to develop a fever. Fever is an inflammatory response that extends beyond the site of infection. The right ratio of ingredients like inflammation, pyrogen is necessary to develop a fever. Like that, good health. Many people who have infections in the body do not develop a fever. Many people who have a diseased organs may not develop a fever. This is the lack of right ratio to development of fever. How can create a fever? Hyperparesia and fix within hours. I can, 1 second. The base of one of the criteria of a scientific discussion is to reproduce. I can make a fever within hours, and I can also cure fever within a half year or more. I can easily make a fever in tonsillitis patients within an hour with the help of ice water. I want to discuss with you in people with tonsillitis, children with elderly using ice water, we can create a fever and hyperparesia within a few hours. With the continued use, we can cause death in them. Thalekuthal is the best example. Thalekuthal is the traditional practice of senticide, killing of the elderly, involuntary euthanasia by their own family members, observed in some parts of southern districts of Tamil Nadu, state of India. This is the process in which an extensive oil bath is given to an elderly person. before crack of dawn. He or she is given several glasses of cold water, cold tender coconut water, and a head massage with cold water. By doing this, the temperature of the body falls suddenly and causing high fever, which leads to death of the old age person within 1 or 2 days. Do you see any difference between fever in viral diseases like the present COVID-19 or in Talaikutal? If there is no difference, then why not use the same science? Necessary ingredients to cure a fever. For curing fever, we should know what are the ingredients necessary to develop a fever. Disperse of correct ratio ingredients of fever will decrease the intensity of fever. By removing correct ratio ingredients of fever will cure fever. The purpose of temperature of fever. That is a subject. When the disease made by the virus, bacteria, fungi, venom, horror seed, and horror dream, etc., becomes a threat to life or organs, blood circulation decreases. Temperature of fever will emerge to increase prevailing essential blood circulation, and it acts as a protective covering of the body to sustain life or organ. It is an adaptation. In all diseases which decreases essential blood circulation at temperature, fever will emerge to increase essential blood circulation and temperature. Talaikutal is the best example. I already told you about what is the Talaikutal. It is also seen in Google also. There is no way other than this to increase prevailing essential blood circulation for a sensible and a discrete immune system to protect the life or organ. Modern science says fever is an immune response against infection. If fever is an immune response against infection, temperature of fever is not a surplus temperature. It is not supposed to be eliminated from the body. If we try to eliminate the immune response against infection, the infection will increase. It is dangerous to immune system. It is necessary to protect the immune system to increase essential blood circulation, never allow body temperature to lose by atmosphere. A brooding hen's body temperature, like that, during fever to increase essential blood circulation in the body, the brooding hen does not eat any food. Brooding hen's food is the heat, which helps to convert fat into energy. Many people have a misconception that fever should be changed or cured. It is not a fever that needs to be changed. It is the disease or cause of disease that needs to be changed. Fever is an adaptation only. Fever can be compared to our soldiers. Sometimes our patriotic soldiers may be defeated, surrendered, or even die. It is not due to the anti-national activity of our soldiers. If fever temperature is removed in Talaikutal, it becomes dangerous. A symptom can never cause fatal. It is said that the temperature above 41 degrees can cause febrile fits and it may cause fatal. New fever definition. If essential blood circulation decreases to organs, fever is a sensible and a discrete action of immune system to increase essential blood circulation as a self-defense mechanism of the body to sustain life or organ. We can answer almost all the questions about fever with this definition. If we avoid or evade from this definition, we will never get a proper answer to even a single question. Why does not everyone with a disease have a fever? Virus, bacteria, fungi, etc. does not cause fever in everyone because the cause of disease does not decrease essential blood circulation to organs, or the body does not need fever protection, or the immune system does not have energy to produce the temperature of fever like in AIDS. How can we prove that the temperature of fever is to increase essential blood circulation? If we ask any type of question related to fever, by assuming that the temperature of fever is to increase essential blood circulation, we will get a proper answer. If we avoid or evade from this definition, we will never get a proper answer to even a single question. If we do any type of treatment by assuming that the temperature of fever is to increase essential blood circulation, the body will accept at the same time, body will resist whatever treatment to decrease blood circulation. Third, if we measure the heat energy used for which activities in fever, we will know the purpose of temperature of fever. No further evidence is required to prove the temperature of fever is to increase essential blood circulation. Let me know new parameters to prove the purpose of temperature of fever. We can prove that the temperature of fever is to increase essential blood circulation with that parameters. Fever is a protector or destroyer of brain cells. Two questions arises. Will temperature of fever protect brain cells by increasing essential blood circulation to the brain, or will it destroy brain cells? Delirious fits formation in disease-related fever. When disease increases, essential blood circulation and energy level also decreases. The vertical height between heart and brain is more than 1 feet. When the disease becomes severe, ability to pump blood to the brain decreases. The blood flow to the brain decreases and delirious or fits are formed. As a result of this, brain cells are damaged. So the patient might be paralyzed or may even die. Fever and delirium. Fainting or delirious or damage to the brain cells is not due to the increased temperature of fever. It is due to decrease in blood circulation to the brain. Without knowing the purpose of fever, it is said that fits or delirious would happen due to increase in temperature of fever. A temperature of 41 degrees never make any problem to our body. During summer in some parts of India, atmospheric temperature is more than 45 degrees. If atmospheric temperature is 41 degrees, there is no such a history of one have been affected with a delirious or fits. A sauna temperature ranges from 160 to 200 degrees Fahrenheit. In a steam bath, a person being 15 to 25 minutes. While taking a normal steam bath, temperature inside the bath is more than 50 degrees centigrade. There is no such a history one had fainted or affected with a delirious or fits. During fever, if we take steam bath, fits never happens. Body circulation never decreases. Skin never shrinks as in fever. During fever, as you know, as you ever, blood vessels and skin are shrinks, blood circulation decreases, and body shivers to increase temperature. If there is 4 degree increase in atmospheric temperature, we have never heard of anyone's blood vessels and skin being shrink, blood circulation decrease, and the body shivered. If temperature is reduced to a normal level, blood circulation will never increase. Fits is never cured without an increase of essential blood circulation. Signs of curing fits. Regaining consciousness even before the temperature subsides. When a fainted patient lies on the floor, the vertical height between the heart and brain decreases. So blood circulation increases to the brain, and so fits is cured even before temperature subsides. Many people misunderstand that regained consciousness is due to reduction of temperature. Shivering is to increase blood circulation. Hyperparesis temperature is 41 degrees centigrade. Only 4 degree increase in body temperature. If 4 degree increase in temperature occurs, there is no history to show that shivering occurs. If disease increases, blood circulation decreases. Shivering not due to increase in fever, but due to increase of disease, decrease of energy, immunity power, strength, essential blood circulation, etc. Shivering helps to increase heat. Then essential blood circulation will increase. Fever and shivering are protective actions according to the aim and target of immune system. If the body produces amount of heat, energy, or force or strength, the body will never shiver. During fever, one shivers due to lack of enough temperature in shivering area. In other words, if a body have enough temperature to maintain its normal body temperature, it will not shiver. The decrease of essential...
Excuse me, Professor.
Yes.
I'm afraid we'll have to ask you to go to the summary because we are starting to run out of time for this segment.
It's okay. The animal behavior. Buffaloes does not try to lie in water, not drink it during fever. Buffaloes manage body temperature by immersing their body temperature in water and drink it. But when they have fever, they do not drink it or enter water or drink it. So some thought-provoking questions. In Talekutal, what are the necessary ingredients to cure hyperpyresia? Is conservative fever treatment suitable to Talekutal? Do you see any difference between elevated temperature in hypothalamus in viral diseases like in COVID-19 or in Talekutal? If there is no difference, then why two types of signs to reduce temperature or fever? Ways to eliminate fever by increasing blood circulation. We just listen to the messages from the body and act accordingly. Increasing essential blood circulation, right treatment for fever. So various types of increasing methods. Blood circulation never increases without increase in temperature. Fits can be cured without increase in essential blood circulation. Apply extra heat from outside and inside to the body. It will help to immune system to fight against infection. To increase essential blood circulation, never allow body temperature to lose by atmosphere and apply extra heat from outside and inside to the body. It will help to immune system to fight against infection. Hot blanket, steam bath, hot sandbag, or thermal pad, hot pepper water, hot drinks can be used. Mechanism application of heat. This is the most important. When the temperature produced by the body due to fever and heat which we applied on the body combines together, the essential blood circulation increases. Then immune system will stop to produce heat to increase essential blood circulation, and the body will get extra heat from outside and inside without any usage of energy. Heat is the fast and efficient remedy to reduce inflammation and increase blood circulation.
Thank you.
So, okay. Now we can say so many type of treatments like an upset. When we apply heat from outside and inside to the body, all the signals, symptoms of fever, like and upset, the hotness according to the directions commands the immune system. If fever is immune response against infection, our body like and upset at the heat. Now the role of sponging. Whatever happens to a brooding hen and its egg, if sponged with water, the same happens to a fever patient. Like in COVID, if temperature is decreased by sponging. If fever is immune response against infection, our body dislike and hate temperature reducing methods. If fever is immune response against infection, our body like and upset hotness. If fever is immune response against infection, temperature of fever is not a surplus temperature. If we reduce temperature by water sponging, infection will increase and it may be dangerous. That is happened in COVID-19. It is not part of disease, it is a part of self-defense mechanism of the body, and it is adaptation. A disease does not require fever, but at the same time, fever requires a disease. For the disease and the fever are different, we cannot include body destructing disease and body protecting fever in same group. So the research, I have so many evidences to present a system of fever treatment. I am thoroughly against the present system of fever treatment. It is proved that so all the conservative fever treatments not only does not make a radical cure of not only does not make a radical cure of the feverish condition, but also further decrease of essential blood circulation and increase inflammation, and it leads to the body to more dangerous. If temperature decreases by sponging or paracetamol, disease or cause of disease or cause of fever is never decreased. It is proved that fever medicines are more dangerous than disease, and fever treatments are more dangerous than medicines for fever. A lot of problems emerge not knowing the purpose of fever.
Excuse me, but I am afraid we're really running out of time. We've already regrettably had to probably skip the question and answer session for this first group, but we'd really like to also give a chance for the following.
Thank you.
I'm really sorry, but here's hoping that we will still have a chance to listen to this in full, perhaps on our next conference. And right now we'll connect with Professor Shishir Gokhale from the Manipal College of Medical Sciences, where he will talk about the preparation of the vaccine for the COVID-19. Professor Gokhale, can you hear us?
Yes, sure. Okay. Am I audible?
Yes, so please welcome and please take the floor.
Okay. Thank you. May I start?
Yes, please go ahead.
Thank you. Good afternoon, everybody. At the outset, I would like to thank the organizers of this conference for the opportunity to be with you all on this very interesting and very captivating topic about COVID. Here you can see two slides, two photographs, maybe of the same place. One before 2019, when the life was full of bustling, we had freedom to do whatever we could. And then came in COVID and put us back into our houses with 1001 restrictions on so many activities. Wouldn't we like to go back where we were before 2019? Of course, all of us would. But is it possible? Perhaps. Yes, perhaps if we have a successful vaccine which can take us to that place. So let's have a look. Where do we stand? Where we can go? And what can happen? If we have a vaccine against COVID, what we can get? If the vaccine can achieve all these goals which are there, or at least the last 4 goals, if it can achieve, then maybe we can convert this life-threatening disease into a minor inconvenience. Something like a common cold, like all other coronaviruses, and we can get on with our life. Will it prevent reinfection? Well, even if it doesn't prevent reinfection, if it can prevent death and disability, we are still happy. But are we going to reach there? The international efforts and collaboration, work by thousands of scientists day and night and huge investments, have seen some wonderful results. In a short span of 9 months, from the identification of the virus, we have come to a stage where we have got vaccines in clinical trials and many of them ready to be launched. Well, huge progress, huge promotion. For these vaccines, which there are more than 200, variety of technologies have been used. We have used conventional technology with killed viruses, live attenuated viruses, or subunit viruses to create vaccines against a large number of diseases. Yes, these techniques are not foolproof. They got their own disadvantages, but at least we know this devil. We can overcome the disadvantages and we can be careful about what we are doing. The COVID has given us a chance to explore newer technologies, mRNA vaccines, DNA vaccines. Well, these methodologies may overcome some of the disadvantages of the conventional techniques, but solution to a problem is always beginning of a new problem. So these newer technologies may lead us to some areas where we might be facing unexpected results, unexpected consequences, and sometimes some complications. How will the vaccines be delivered? Well, we have these 4 or 5 conventional courses in which it can be done, and how the vaccine will stimulate our immune system and what will be our response depends not only on the type of vaccine but also on the mode by which it is transferred to us. We can have systemic immunity with IgM and IgG, which can protect us against circulating virus but may not protect us against an invader. On the other hand, if we have the local immunity with IgA antibodies, we can stop the enemy at the gates. So perhaps we need a combination of both local immunity as well as systemic immunity to protect us from COVID. The rush and the warp speed operations have brought us to a level where we are in phase 3 human trials. In fact, some of the pharmaceutical companies have gone into commercial productions and stockpiling the vaccines, just waiting for the green signal to launch it. Russia and China have perhaps gone a step ahead. They have taken an ambitious leap by deploying the vaccines in large number of population. We still do not have much data on it. So we need to be careful while assessing whether these vaccines will be useful, not useful, or sometimes even harmful. The question comes is, will the vaccines work? Well, the vaccines can lead to immune system stimulation in our body. But the pathogenesis of COVID-19, as has been brought up earlier, and some of the speakers will be bringing up further, my colleague Dr. Naik will speak about it, is primarily understood to be an immune response and a cytokine storm. That means the COVID-19 has got more severe course, more morbidity and more mortality when the immune response and the cytokine storm is very high. All this data of not effective plasma therapy and not lasting the antibodies beyond few months even in recuperating patients reflects that the antibodies perhaps may not protect us, but on the other hand may make us more susceptible to the severity of COVID. Now here comes a question. The vaccine can stimulate immune system, but will this immune system aggravate the COVID or will it protect us against COVID? We have seen antibody dependent enhancement of disease in dengue, and will something like that happen in COVID? If so, it's perhaps better not to have a vaccine than to go for a vaccine. But this question cannot be answered today till we actually see the effects of vaccine. We had number of successful vaccines against measles, mumps, rubella, tetanus, and we have almost eliminated those diseases. We have some vaccines where we had partial success like influenza, where we need to reconfigure the vaccine almost every year to give some amount of protection, not 100% protection. On the other hand, dengue is a disease where vaccine can be given only after the person has already been exposed to dengue. All our scientific advances have not always generated good effects because we still have been unsuccessful in many cases in generating vaccines. What can we bet for COVID vaccine? Should we bet on a vaccine which will produce systemic immunity with IgM and IgG response? Well, perhaps yes, but also needs to keep in mind the pathogenicity of the disease. Perhaps I would like to bet on a vaccine which can produce local immune response so that the virus is stopped in its tracks right in our nose or throat, and no further immune stimulation, no further inflammation, no further complications of COVID. Maybe we need a vaccine which can stimulate the cell-mediated immunity and not the humoral immunity alone. Or perhaps a vaccine which can combine effect humoral and cell-mediated immunity. But going back to the pathogenesis, if pathogenesis is based on immune storm, cytokinic storms, then maybe we should be looking for an immunomodulating intervention, something which will stimulate the T cells, which will suppress the antibodies response so that we can have more control on the clinical outcome of COVID, not only protecting but also preventing the deaths. Maybe BCG not directly affecting virus, but by stimulating our T cell response can do that job. So where are we today? Well, the experience with other coronaviruses has not been very good in terms of vaccines. Will the story be different for novel coronavirus? Only time can tell. Even if we get a vaccine, perhaps we'll need multiple doses. Most of the vaccines which are in trial require at least 2 doses to give certain amount of immune response. But do we require same vaccine for everybody, or will be using different vaccines for different groups? And how long this vaccine will protect us? Will we need a vaccine every year like influenza, or will we get a vaccine which will give us an immunity like against mumps? It depends on viral mutations. One of the speakers has emphasized on which clades are getting more prominent. Will they further mutate? Will they further change? Well, only time can answer these questions. And in the timeline, we have nothing on the horizon till the end of 2020, because even if even if vaccine will be available by end of the year, it still will have to undergo lots of efficacy, safety, and follow up to give us a vaccine for use for general population. I don't think before 2022. So the question comes, which is in mind of me and which everybody else: when and will I get a vaccine? Well, it depends on international and your local criteria. Obviously, the priorities will be given to the frontline workers like the healthcare workers or the people who are more likely to be exposed to COVID, and then perhaps to the susceptible population, the elderly ones who are bearing the brunt of the disease, and then only will be you and me in line. So let's wait for our turn. It's not going to be so easy. These vaccines are not going to be magic bullets. It could be years before they become available. And even if they are available, can we afford it? These are going to be expensive vaccines. The COVAX scheme of countries have proposed that they will make these vaccines available even to the developing countries, but in limited number, to cover almost 20% of the population. Will that be enough? No. A vaccine which covers only 20% of the population will not generate herd immunity. We need a coverage of about 60 to 80%. We have just seen this happening with measles, where the vaccine coverage came down to 50% and below, and we had outbreaks of measles. So we need to have a vaccine which will be available for 60 to 80% of the people, where it could protect them. Then it can lead to herd immunity, and then only we can expect some degree of normality coming into our life. Well, let's not forget about the anti-vaxxers. I just now got a call a little bit ago from Elon Musk, who has declared, I am not going to take vaccine. Well, he is not only one, there is a large number of people anti-vaxxers. So where do we stand, ladies and gentlemen?
Excuse me, Professor.
Knowing is enough, but yeah.
Because we have already
So till we get a vaccine,
overrun our time for this
till we get a vaccine,
segment, if you could reach for
one
the
second.
summary.
We should not wait for vaccine. We should continue with our protection. We should continue with our activities and protect ourselves and population. Thank you very much. Have a nice day.
Thank you, and sorry again for rushing you, but I'm afraid we've already run out of time for this first group. I'd like to once again thank everyone involved for participating and for very illuminating presentations. But now it's time for...
Yeah.
Oh, we do...
Okay, should I start?
We do still have one more expert ahead of us. Please, introduce yourself and go ahead with the presentation.
Okay, good afternoon, everyone. Myself, Dr. Prashant Kumar from Ahmed Institute of Virology and Immunology, Ahmed University, Uttar Pradesh, India. I am basically a medical virologist and mostly I work on the respiratory viruses. Previously, I've been working on various different strains of influenza A virus and the development of immunogens against this virus. So now at this time of COVID-19, we have also been trying to do some preliminary studies for the start of the development of an immunogen against SARS-CoV-2 virus. So here on this platform, I will be discussing the scenario of COVID-19 in India and thereafter what all management practices are there. So to start with, first of all, I will discuss about the current scenario of COVID-19. In India, actually, the total number of cases as on September 30, today, has crossed about it's more than 62 lakhs as of today, and the number of total number of death cases have been reported nearly more than 90,000. Since until yesterday, the approximate number of cases that were reported on daily basis was approximately 70,000 to 80,000 cases were reported. No doubt the number of deaths due to COVID-19 has decreased, but it's still worrisome. We can't avoid it, avoid looking at it. So there are a number of management strategies to have a control on this COVID-19, and it's spread across the country. And I will be discussing the management practices that have been practiced by our government to take care of COVID-19 in India. Mostly, as has been discussed by other eminent Indian scientists also, since the start of this pandemic in India, in March 2020 itself, the practice of social distancing is being maintained. Our government imposed lockdown for a period of more than 2 months like that, and we have been seeing that when there was lockdown, the number of cases were either constant or they were decreasing. But still, when there was unlock period, the social distancing needs to be maintained so that the spread of this SARS-CoV-2 among the population decreases. Actually, in India, the population varies from one area to another area. The population density actually varies from one area to another area. And based on the population density only, we can say that social distancing is very, very much important. So not only this, it's very important to have extensive practices so that the people in different areas may be made aware of the importance of social distancing and of the importance of wearing masks. Because many a times people think that, okay, it's COVID-19, even if they are infected, they may get cured on its own. But they should be made aware that it's very important for them to wear masks and get tested so that others may not be infected because of them. It's very important to control this disease. So many a times the people are very much ignorant about it. They are very much reluctant for this. They don't care for others. So extension services are very, very important for this purpose. Now, coming on to our government's other steps to manage COVID-19 in India, in today's scenario, the diagnostics tests is very important. The number of tests that are being done on daily basis is to be increased significantly. As of now, nearly 1.2 to 1.4 million tests are being done every day. And this has increased from about 10,000 in early period like March, April, then today it's about 1.2 to 1.4 million. So there has been significant increase in the number of tests, and it gives an idea that what all populations are affected and what all management practices needs to be done. So in India, we are going for RT-PCR based test. We are going for antigen-based tests. Serosurveillance is also going on. So these tests are being done regularly in India. So and it's very important. Rather, again, the awareness needs to be provided to the people, so that even if they are suffering from a mild disease, they must get tested, so that if they come to know that they are positive for COVID-19, they may take utmost care to quarantine themselves so that the disease is not spread because of them. So this practice needs to be done here. Now, coming on to the research that is being done on this COVID-19. Before research, I would also like to talk about the readiness of the various hospitals and healthcare practices. Actually, our government has also taken various steps to increase the number of beds available in the hospitals, so that in case of an outbreak, this outbreak of the disease in a new population, quarantine facility may be sufficiently available in various areas. So this is also very important, and we have taken steps. Our government has taken steps for this. Now coming out to research on vaccines, everyone has been talking about therapeutics or vaccines, which is very, very important for the control of this. Actually, all the offices, all the schools are just waiting for the vaccines to come so that they may be opened. So today in India, approximately 7 to 8 vaccine candidates are under research. And as we all know, 2 vaccine candidates are already under phase 2 clinical trial. And we are waiting for them to get cleared. So once the vaccine is available, for given to the common population, we may afford, our government may afford to open the offices and.
Oops, it seems we've lost the professor here. But let's hope this is just a minor connection issue that will be resolved in a second. As I'm really looking forward to hearing the end of this speech. Well, let's hope we manage to reconnect soon. Or if not, we'll then probably have to go on a short break before we reconvene with the next group of experts. Regrettably, as we've already run about 15 minutes past our schedule, we'll most probably have to skip the question and answer session for this group. However, we will do our best to make sure that every speaker will still have chance to present their findings, no matter the time. Well, let's I'm afraid that we've lost this connection irrevocably. However, we are still having a chance to connect with Professor Ahmed El-Kafrawi from the Al-Azhar University, an accomplished Egyptian scientist whose main scientific interests are vector-borne disease virus and hepatitis viruses. Dr. Kafrawi is currently actively researching COVID-19, and he will be telling us about the pros and cons of the main, most popular approaches to drug development in case of COVID-19. As far as I am aware, his speech will particularly focus on the preclinical and clinical testing stages of drug development. Let's wait for Professor Kafrawi. Oh, on the other hand, we've got an update to take a short break, about 5 to 10 minutes before we reconvene, and hopefully we'll manage to get all of our connections in so we can proceed smoothly. See you in 10 minutes.